Hello friends, I am Dr. Prashant Sharma and you are watching MEDICO'S Hub. In this video, I am going to discuss the structure of neuron. Neurons are the longest cells of human body and these cells never divide. Only these can be repaired in the peripheral nervous system. Once a neuron is destroyed, it is replaced by the neuroglial cells only. A neuron cannot regenerate another neuron. The metabolic process or the metabolism in neurons is very slow and it uses about 70% of energy in maintaining the activity of sodium, potassium, ATPase pump. Now, the structure of neuron. The typical human neuron is multipolar. That is, it is having many processes. This is the axon part and this one is the cyton, which is having the nucleus. Many branches emerge from the cyton part which finally divide to form fine branches. These branches are basically termed as dendrons or dendrites. The terms dendrons or dendrites are basically used synonymously. Now this is the cyton part. This is cyton, or it can also be termed as cell body or soma or perikaryon. The structure of cyton may be stellate, fusiform, pyramidal, or round. These branches which emerge out from the cyton are termed as dendrons or dendrites. This is nucleus. And around the nucleus there are some basophilic bodies made up of aria which also extend into dendrites. These are known as Nissls granules. These are Nissls granules. So these are the site of protein synthesis. These are aria. In addition, the cyton may also have mitochondria and the Golgi bodies. Now, these are the dendrons or dendrites. This one is cyton or cell body. If the sectioning is done or poisoning occurs, then these Nissl granules degenerate to fine powder and then disintegrate completely, which is known as chromatolysis. Now, this cyton becomes narrow and continue as axon. This part of cyton which becomes narrow and continue as axon is axon hillock. Axon hillock lacks the Nissls granules. Nissl granules are absent in axon hillock. Now, I am drawing another neuron. This is another neuron. Then this axon is dividing into smaller branches. These branches can end on the dendrites of another neuron or on the cyton of another neuron, or these may end on the axon terminal of another neuron, or these may end on muscle also, muscle cell, that is sarcolemma of a muscle cell. This is a muscle cell. So, first of all, the remaining part of neuron is axon. Axon is dividing into many branches which are termed as telodendria. These all are telodendria. Telodendria are having the terminal portions, swollen portions. This swollen portion, terminal swollen portion, is known as synaptic knob or terminal buttons. So this is synaptic knob or terminal button. When this terminal button ends upon a dendrite, it is exodendritic synapse, which is most common type of synapse. And when it ends on cyton, it is exosoma, that is exosomatic synapse. And this type of synapse is exonic synapse, and this is what we say neuromuscular junction. Now the axon is covered over by Schwann cells in PNS. So this is Schwann cell. The plasma membrane of Schwann cell is neurilemma. Schwann cell wraps the axon just like crab bandage. It may wrap up to 50 to 100 times around the axon. And the region between the Schwann cells is known as node of Ranvier. So this is node of Ranvier. While the part of axon between the start of myelination and the axon hillock is termed as initial segment. Now if we compare the initial segment with node of Ranvier, then length is 10 to 60 microns, while the length of node of Ranvier is 1 micron. Voltage gated sodium channel density, it is 350 to 500 per square micron. while here it is 2000 to 12,000 per micron square. Initial segment is the site for initiation of action potential in motor neuron, while the node of Ranvier is the site for initiation of action potential in sensory neuron. So this is the comparison of initial segment and the node of Ranvier. It's important to discuss two more terms. One is neurite. Neurite means the processes of cyton, that is dendrite and axon. Another term is neurofibrillar network or neurofibrillae. Neurofibrillae basically involve neurofibrils microtubules neurofilaments, which are intermediate filaments, and the microfilaments. So these all are neurofibrillae. Clinical importance is that in Alzheimer's disease, the proteins involved in the formation of neurofibrils are altered and neurofibrillar tangles are formed. So this is the structure of neuron. Hit the like button, share and subscribe our channel to get the latest updates and notifications.